So as far as I remember, last time we have finished with the discussion of anatomy of the muscle, and I want to remind you just a few things about it. You have a muscle as a whole surrounded by epimysium. Muscle consists of multiple fascicles, and each fascicle is surrounded by perimysium. And inside of the fascicles, you're going to find the muscle fibers, okay, the cells. Each fiber is surrounded by endomysium. And endomysium and sarcolemma, the membrane of the muscle cell, are two different things, okay? Now, if you would look deeper into the skeletal muscle fiber, you will find that it contains numerous myofibrils, rod-shaped structures that in turn consist of individual units called sarcomeres. And inside of each sarcomere, if you look deeper, okay, you will find a number of filaments. Those filaments, we call them filaments, are actually proteins. Okay? So myosin, which composes thick filament, is the protein. Actin, which composes thin filament, is the protein. Elastin is the protein. All those structures inside of the sarcomere proteins. And I want to remind you also there are storage units, glycosomes, that store glucose, because muscle requires enormous amount of energy, and protein myoglobin that stores oxygen, since muscle needs a lot of oxygen, for its metabolism. Now, let's talk about sarcomere in the time we have left. Try to carve out all the time that we can. So this is the smallest contractile unit. Okay, you can see one sarcomere, another sarcomere. Think about them as the cars of the train. Okay, so on the sides of the individual sarcomere, and this is, of course, this is the schematic representation. Okay, so this middle picture is the schematic representation of the sarcomere. Sarcomeres are bound by Z discs, which are protein structures. They connect sarcomeres to each other. Okay, and two major contractile filaments, thick filaments, the red ones, the myosin filaments, and thin filaments, blue actin filaments, are attached, physically attached to Z discs. Actin filaments are attached directly, while the myosin filaments are attached via the protein titin, okay, which forms elastic filaments. So you can say, the names of the filaments are pretty descriptive: thick, thin, elastic. Okay. Now, myosin filaments here, they form the part of the sarcomere that is called the A band. Actin filaments form two parts of the sarcomere. It's A band and so-called I band. Now let's stop for a second and just look at the picture. Look at the picture. So this part here, okay, filaments overlap. Does that make sense? Can you see them overlapping? Since they are overlapping, that part of the sarcomere is going to be darker. You can see it right here. Do you see that? In the places where I bands are only actin filament, only actin, okay? Like here. Only actin filament. There's no overlap, and this area of the sarcomere is going to look lighter. Now, do you remember the word striations? When you look at the skeletal muscle, you see that skeletal muscle is striated. So the dark A band is a dark striation, and the I band is the light striation. That's why skeletal muscle appears striated. The molecular structure of the muscle fiber, of even myofibril, determines the appearance of the muscle, determines the striations. Does that make sense? In the center of the A band, you have a very narrow, practically invisible light area, which is called H zone. Okay? Let me erase on top. So you see that little portion here? This is H zone. This is the area where myosin does not overlap with actin. You can see only myosin here. Only myosin. Right? In the center of H zone is M line. It connects different myosin filaments, and M line consists of the protein called myosin. Holds the filaments together. Now, actin filaments, as I mentioned, are connected directly to Z discs. Myosin filaments are connected via elastic filaments that consist of the protein titin. Does that make sense? Now, different filaments of the sarcomere are attached to the membrane of the muscle cell, to the sarcolemma. This attachment is provided by the protein called dystrophin. And as you can guess, in a condition called muscular dystrophy, this protein is destroyed. So integrity of the muscle fiber is altered. Okay. Now, nebulin is the protein that is not shown here. Regulates the length of actin filaments during the assembly of the actin filaments. And it also inhibits the ATPase, the enzyme that is found inside of the myosin filaments, inhibiting muscle contraction. Now, this element, A band, I band, M line, Z disc, elastic filaments, thick filaments, thin filaments, they may not make sense until you look at the mechanism of the actual muscle contraction. For this, we need to figure out the structure of the thick and thin filaments. Thick filament consists of many myosin proteins. That's why we call it myosin filament. Each myosin molecule, you can see it here at the bottom, consists of two heavy, the tail parts, and four light head parts, chains. Okay, so think about this. It can be presented something like that. Okay. So that's heavy, and those are light fragments. Okay. So tails of the myosin filament, tails of the myosin protein, they actually form the filament. So this rod-shaped structure, can you see that? Like, looks like stem. Okay, that huge cylindrical structure in the center is composed of the tails. These protrusions are heads of the myosin proteins. And these heads have two very important functions. Function number one, heads of the myosin protein can bind to actin. Does that make sense? Number two, they can break down ATP. And ATP is the molecule necessary for the contraction since it provides energy for contraction. Okay. Now, actin, which forms the light chain, okay, the thin filament, okay, consists of multiple globules. So those are molecules of so-called globular actin. And think about this. Those molecules of globular actin, they attach to each other and form something like a chain of beads. Okay? And this chain of beads, this blue filament is called fibrous actin. Okay? So interaction between myosin and actin can be shown like this. My palm is the head of myosin. The ruler is actin. The rest of my arm is myosin. Does that make sense? That's how they interact. So my arm is myosin. My palm is the head of myosin. This ruler is actin. Normally actin is bound to two more proteins. This long beige, I don't know, whatever you want to call this color, beige line represents the protein called tropomyosin. Tropomyosin, in fact, prevents interaction between myosin and actin. And troponin, those yellow molecules, is the protein directly responsible for the initiation of the muscle contraction. It binds calcium. Does that make sense? And once it binds calcium, the entire contraction occurs. Now, to make sense out of the sarcomere, what I want to talk to you about in like a few minutes, and we'll discuss it later a little bit. What I want you to understand is the basic, very, very basic mechanism of contraction. Look at the sarcomere. When myosin heads, look at my palm, start to bend, actin slides towards the center. Can you imagine this by looking at the picture? Okay, actin slides towards the center. So Z discs move closer to the M line. Myosin doesn't go anywhere, just stays in place. If I'm myosin, I'm not going anywhere. Do you see that? While actin slides towards the center. So that brings Z discs closer together, elastic filaments shorten, and sarcomere altogether shortens as well. So since each fibril consists of many sarcomeres, when they all shorten, the entire fibril shortens. Do you understand what I'm trying to say? when the entire fibril shortens, the muscle cell shortens. When many muscle cells shortens, you get muscle contraction. Does it make sense? This is called the model of sliding filaments. Okay? Sort of to make sense. And you can imagine when there is a relaxation, myosin sort of releases actin, and Z discs, they move back because of that elastic action of elastic filaments. Does that make sense? So muscle can contract, but when it relaxes, it recoils back. Okay? So I'm going to meet you on Thursday. We will continue with the physiology part.